Two, one. Awesome. We're here, everybody. Well, a huge welcome into all of you. Good afternoon. I know a lot of our folks are joining in California today. I'm in Newfoundland on Canada's East Coast. Abby's in Antarctica. The mere fact that such a thing is possible is sort of outlandish. I think, back a few hundred years ago, this was like something that you could be accused of witchcraft for being involved in a live stream like this. So it's always a pleasure to get to talk to amazing folks in Antarctica. And as many of you may know, Abby has done some really incredible work with her team diving in a lake in the heart of Antarctica. If there's a cooler gig in science than that, I do not know what it is. And she invited me to help host today. We got a chance to do a broadcast with her and her team live in Antarctica a few weeks back, highlighting all the amazing work that they were doing to like 500 plus kids all across North America. And it's just a thrill today to get to be here to introduce some of her stuff to you, UC Davis audience. So I look forward to all your questions. And Abby, if you want to dive in and take us away, literally and figuratively, and highlight all the cool stuff you and your team have been up to, you are good to go. Take us away.
Thank you, Jesse. Hey, everyone. There's a bit of a delay on my end, so apologies in advance if that's a little awkward, but here we go. Jesse, can you see my screen now if I play it?
I can indeed. We've got an epic Antarctic background. You're golden.
Yes, hopefully. I think that's a go. I'm gonna go now. Hope you can interrupt me if you can't see something. All right. Hey, everyone. Thank you so much for joining. It means so much to myself and our team here that you've all taken the time to join us today live from Antarctica. So we are in Antarctica today as we speak. I know it doesn't really look like it, this background. We could really be anywhere in the world. As Jesse mentioned earlier, we got to do a really cool outreach event to some students back at the beginning of the year from our field site, Lake Frixel, which we'll be talking about today. And I had glaciers behind me and the lake, and it was quite beautiful. So this wall doesn't really compare to that, but you should know that there are penguins right outside this building on the sea ice, and we saw some orcas the other day. So it's a bit of a trade-off, but still equally awesome. So yeah, as I said, our team is here with me right on the other side of my computer, and you'll be hearing from them later on in the talk. But I am going to get us started here and introduce you to our work and our life in Antarctica for the last 2 months, what we've been up to, the beautiful sights, and what it's like to dive in Antarctica as well. So for those of you who do not know me, my name is Abbey Dias. I am the University of California, Davis Assistant Diving and Boating Safety Officer. I'm based at Bodega Marine Lab. So hi to all my friends who are watching at the watch party at BML. Thanks for joining in. But my role serves all of UC Davis, and I love my job because I get to work on some really cool projects with lots of different people like this Antarctica project. So this is my second year on this Antarctica project with the UC Davis team. Our project event number is called B047, so that's why you see that by my name. That's a number that the National Science Foundation has given us to recognize our event. And I'm a scientific diver for our team, as well as I focus on the outreach components of our work as well. Hence why I am starting off this presentation for you all. So my goal during this trip to Antarctica was to mainly support science through offering dive safety, diving support resources, actually being a diver and collecting samples, taking photos with my underwater camera and being able to share them to you all, and then also offering some dive training too. So I'm not the most experienced Antarctica diver on our team, but through my job at UC Davis, I get to train lots of different researchers in boating or diving and whatever they need. We also teach first aid classes. And then different groups can come to us with different needs, and we can create courses based on what those needs are for their research. So specifically, the Antarctica team came to us this year for some specialized Antarctic training, and we took the Antarctic divers to Tahoe. So I'm going to show you a little glimpse of that. This was back in September. So you don't get to dive in Antarctica overnight. It takes lots of training and practice and planning to be able to do so. So we took our divers out to a lake that is kind of similar in that it's freshwater to the lake that we're diving in Antarctica. Unfortunately, the visibility in our lake in Antarctica is not the same as Tahoe here, but this is a photo of Tyler Mackey, who is one of the co-PIs on our team here in Antarctica, but is also a UC Davis alum. You'll hear from him later. And this photo, he's demonstrating sampling some Jell-O, which I made to mimic the sampling that we do here in Antarctica of our microbial mats. So you'll hear more about that later, but I love this photo because it shows how precise Tyler is floating above the substrate on the bottom of the lake. And those dive skills were really important for being able to dive here in our Antarctic lakes so that we don't disturb the bottom and we can get some really precise sampling done. So we love Tahoe, but our main goal was to come to Antarctica. So why are we here in Antarctica? Why do we do research here? I love Antarctica because it's designated as the continent of science. So many people think of Antarctica for the ice and the amazing creatures that live here, but it's been dedicated as a place for science and only for peaceful purposes. And that's by the Antarctic Treaty, which was established in the 60s, and it's now been signed by 58 nations. So many people take part in science around Antarctica, and so it's the perfect place for it. And in the summertime, which it is now, so it's winter up in the northern hemisphere and summer down in the southern hemisphere, there are 24 hours of daylight all day, every day during this season. And so in the summer, it's daylight. In the winter, it's 24 hours of darkness. And that requires some really unique adaptations for the life that lives here, as well as the just generally harsh conditions for living on this continent. It's cold, it's dry, a lot of interesting weather patterns and whatnot. So it's a great place to study the extreme life that does live and thrive here in Antarctica. So how did we get here? Well, a couple of months ago now, I guess, I left San Francisco and took a 13-hour plane ride to Christchurch, New Zealand, which is where the U.S. Base is for the Antarctic program, where we get outfitted with all of our special weather equipment and go through some trainings. And then after our time in New Zealand, we take another plane from New Zealand down to McMurdo Station in Antarctica. And that looked something like this. This is the military plane that we flew on. And you can see we are all quite jam-packed in that plane. And the plane ride's interesting because it's pretty loud, it's pretty dark, and you don't know exactly how long it's going to take. And that's because depending on the weather, you might have to boomerang back and you might not make it onto the continent. You might have to come back to New Zealand. A few of our teammates experienced that this year. So that can be a really long flight. But if you're lucky, you might make it there in about 7 hours. And if you do, you land and go to McMurdo Station, which is where I'm talking to you from right now. And McMurdo Station is run by the National Science Foundation, and it's like a small town. It's basically like a college town, a lot smaller than Davis main campus, but it's got dorms, it's got a galley, it's got a waste treatment facility, and so many things that you need to run life down here, and also the science building and research labs that we're in right now. And then once we spent some time out here in McMurdo at the beginning of our trip doing lots of trainings, like how to be safe out in the field, how to work with helicopters, things like that. We eventually took a helicopter out into the field. And here's a little clip of what that flight looked like over the sea ice going to our remote field station. And it's really important for us to know how to work safely around helicopters because they are our support for our entire time out in the field. So they deliver us our food, they deliver us all our safety equipment, things that we need, personnel. So we work a lot with helicopters and they're quite interesting. And when I came to Antarctica, that was my first time flying on one. It was pretty cool. And then we arrived at beautiful Lake Frixell, which is in the McMurdo dry valleys. And so this might not be what you expect Antarctica to look like. You'll notice there's a lot of dirt here. It's pretty brown. That's because the McMurdo Dry Valleys, they are a polar desert. And so this is the largest ice-free zone in Antarctica. But right in the middle here is our Lake Frixell. So you can see beneath these mountains comes a lake, and that lake has water beneath it. And that is what supports life here in the dry valleys. So it's an incredible resource in this area. back when the first explorers came to Antarctica, Robert Falcon Scott, his team ventured into the dry valleys in, I think, 1900s, 1901 or something like that. And they crossed the dry valleys and wrote in their notebook that it was the Valley of the Dead. And they said that there was no living thing that lived here. And I think that's unfortunate that he did not bring his scuba gear or his microscopes, because actually there is a lot of life here, but it just looks something like this. Hard to see with your eyes. It's microscopic. Here's our charismatic megafauna of the dry valleys. We've got nematode worms and also tardigrades. So everyone loves a good tardigrade. Tardigrades, we call them sea or water bears. Yeah, water bears. And they're these really cute little creatures. This is a video that Marisol took, one of our team members, from a sample in the lake, and it's eating. And they're just these really cute bears that are super robust and able to live in really harsh conditions. So that's what we find out here in the dry valleys. So our goal out here for our research at Lake Frixell was to understand how the microbial life persists in the harsh environment and how it responds to change. And so in order to be able to understand this question and begin to answer it, you have to understand how we survive in these conditions out at Lake Frixell. So I'm going to show you what our life looked like out there. We sleep in tents at Lake Frixell. And at first, when I was told that I was going to be camping in Antarctica, I did not think that was serious. I was like, what? You can't sleep in a tent in Antarctica. But in fact, you can, and it's not too bad. It's actually can be really warm in the tent. It heats up with that 24 hours of daylight. So it's quite pleasant. So we sleep out there in tents, and then we have some main huts that support the generators and things that run our life and our food. So this is what the main hut looks like. This is where we have dinner at Lake Frixell Camp. Sometimes we can have a lot of people sharing one space at the same time. And it's also where we can do our science work, have our science conversations, do our planning, answer our emails when the Wi-Fi is good. And this is what we eat. So in order to be an Antarctic explorer, you have to be willing to eat frozen food and dry food. And we were very fortunate to have an amazing camp manager named Taylor, who was an awesome chef, and she made the best food out of some pretty limited resources, I would say. And yeah, we were eating great out there. So it's really not too bad either. So those are the ins of Lake Frixell, but the outs of Lake Frixell Camp, that's what this looks like. Our toilet situation. You can see there are 2 toilets here, and this is not for buddy bathrooms, but because we have to separate our solid and our liquid waste, and then we have to ship it off continent. So you can see there's 1 toilet for liquid, 1 toilet for solids, and my favorite thing about this is the foam toilet seat, because it makes waking up in the morning to a cold bathroom not too bad. So that's actually a really important part of life in Antarctica, even though it is maybe a little bit silly. But because this continent has been designated for science and peaceful purposes, it's also highly, highly protected for environmental purposes. And so we leave no waste. We practice leave no trace principles, and we basically everything we bring in, we pretty much ship out off continent so that it doesn't disrupt the environment and it can stay well preserved. And here is our office. This is what a day in the office looks like out at Lake Frixell. This is out on the lake right in front of our camp. And this is the dive hut and the dive hole. So in order to get through the office door, we have to break through every morning. I don't know if you can hear that chipping. Every morning we chip through that layer of ice on top of the ice hole in order to get down to do our dives, which is how a majority of our science happens, or our samples are collected and then science happens on them later. So that's a huge part of our work here, is to be able to do the diving. So our team members spent 28 approximate hours melting this hole through the ice. The hole is about, the ice on the lake is about 4 meters, or like 12 feet thick. So it's quite thick. When you go down the hole, you are descending through all of that ice. And if you can imagine, if you're in a regular sized room, it's probably about as high as the ceiling. For those in the at BML who are in the lecture hall, it's not quite that tall. But in a normal sized classroom, that's how much ice is on top of you. So it takes a lot to be able to get through it. But once we have that hole and all of our equipment is ready and set up, then we're able to dive. So here is me in my dive gear. And so, like I said, my main role on this project was to offer diving support in the form of safety and science. And so, I got to do lots of dives while here, along with some of our other team members who are divers. And so you'll see we are connected through this umbilical cord and using a different type of diving setup than I normally use back home on scuba. This is surface supplied diving. So that's why we have that weird face mask looking thing going on because it's a different kind. So this is really important because that umbilical line supplies us with air. So surface supplied diving implies that your air comes from the surface and is controlled by those who you trust on the surface that are taking care of you. It also offers a lot of redundancy in the system. So it's an incredibly safe system. You have lots of different sources of air. So it's great for diving in remote places. It also offers communication. So another one of these lines wound up in this umbilical is our comms line, and that allows us to talk underwater to the folks on the surface and vice versa, which is a really cool plus for doing underwater science. And the umbilical also allows you to find your way back to the hole, which is incredibly important, especially when visibility is not that good. You can always make your way back out. So I'm going to take you for a dive. And first, I want you to focus on your breathing for just a minute. So when we're diving, we care a lot about breathing, not holding our breath. So I'm going to slow down, think about my breathing too, be calm, be ready to breathe slowly. And these organisms that I'm about to show you, they are responsible for creating the oxygen just like what we are breathing. And we're going to dive into the science with our scientists here in just a bit. But I'm going to show you what it's like diving under the lake. So here we go. In the lake. Oops. So what we're looking at here are mats of microbes, lots of tiny different organisms, bacteria, diatoms that are all held together, and they photosynthesize when there is sunlight. And so you can see they're all kind of like lifted off the bottom in these really interesting structures, and that's because when they photosynthesize, they produce oxygen. And that oxygen accumulates into bubbles within the mat, and it basically buoys the mat up when there's enough of the bubbles, buoys the mat up, lifts it up off the surface, and you get these really interesting lift-off structures. So we're going to hear more about that from Marisol. But Lake Frixell is really unique because it's undisturbed. Unlike any other bodies of water that I've ever dove in my life, there's no predators here. There's no fish. There's no sharks. Anything to disturb this life that's here. So it's really delicate and fragile because, like I said, it just takes like couple of bubbles to be able to then lift that really fragile mat up into these structures. And so it's really just divers that are in this lake that are moving around. And so these types of microbes, they grow all around the world, like in the ocean and lots of other places, other lakes, but those places are really dynamic. And so they don't have a chance to form these structures like they do in this lake. And so it provides this really unique study system to be able to look at this kind of life. So here is a photo of one of those structures in the lake. And as a diver, I get to be the eyes of the scientists that are up on top. So not all of our scientists are divers. And I guess not all of our divers are exactly doing science like myself. And so I get to really describe what I'm seeing down below in the lake to those who are on the surface, taking notes and thinking about what kinds of samples they want and trying to get an understanding of what is going on down there. So I get to describe what I'm seeing, and I have to use really precise language and sometimes creative language to be able to describe in detail what we're looking at when they can't see it from the surface. Sometimes I get to bring my camera down like I did for these photos, but a lot of times it's just me talking to Marisol or Dawn on the surface, who are scribbling notes of what I'm saying. So I'm going to demonstrate for you what that's like. So if I was down underwater and I saw this structure, which I thought was particularly nice and why I took a photo of it, I would describe things like, well, there's a thin layer of gelatinous goo on top of it that's kind of coating the whole thing. And then underneath is this sort of light brown mat, maybe a chocolate milk color. And then there's bubbles that are accumulating in the mat as well. I'm noticing that there are some larger bubbles that are up at the top of this structure. With our scale bar in this photo being 10 centimeters, I could estimate that maybe the structure is about 15 centimeters tall, lifting off what looks like maybe a rock beneath. So there's bubbles throughout. And I think I called this slimy fingers on a rock. And I used that to communicate to the team above that this is what I was taking a photo of, so that when I went back to look at my photos, we could find this and know what I was talking about. So slimy fingers on a rock is another creative way that we get to, describe what I'm seeing to those below. So when we did our presentation for the students back at the beginning of the year with Jesse, I got to ask the students how they would describe something like this. And we got lots of really funny and crazy answers. So if you're on YouTube or you have access to the comments too, feel free to write in the comments how you might describe these things as we go through. So I want to appreciate the different bizarre, interesting forms of the microbes in the lakes, the mats, and how they change throughout the lake too. So we're going to look at the different forms of the microbes, and then we'll get into hearing from each of the scientists on the team, and they're going to explain a little bit more as to what kind of questions they're asking about the mats and maybe why some of these things are happening. So this photo was from the shallows, probably about 6 meters deep. 5 meters deep, I guess, as we go down. Forgot I put these on there. As we go down, this is from about 7 meters deep, and this is another form of that lift-off mat. So when it is lifted up off the substrate, we just have a general term for it, which is lift-off, but specifically, this is in dome form. So I could convey this information back up to the surface. There's no scale bar here, but if I had to recall, it's probably about like 20 or 30 centimeters high here. So that's about the size of the things we're looking at. But the liftoff can also get quite big. So as we descend a little bit further, 8 meters, here we go. This is a larger liftoff structure. And this is kind of fun because it looks a little bit spooky and ghoulish. So that laser, if you can see those 2 red dots, that's our scale bar. It's about 10 centimeters between the 2 dots. It kind of looks like eyes on the mats, which makes for some really fun pictures. But the liftoff mats can get pretty large. As we descend further, we get into this type of mat. And this is what we call honeycomb because it looks a lot like honeycomb. The students that we asked earlier, they said it looked like cheese gone wrong. And I love that. So we might use that in future years describing this type of mat. But yeah, this is all within the same lake, within a very small area. Just so many different forms and so many different things to look at. So that's 9 meters. And our scale bar there is about 1 centimeter for those little cubes. As we descend even further, this is about 10 meters deep. We get into a totally different area. So again, all of these things are really close by. So many different forms of these microbes and lots of different things to describe to those on top, to do experiments in different areas and take samples. So I'm going to show you a video of what it looks like when you take samples of these mats underwater. So this is me. And if you can hear volume, you might be able to hear some of my communications from underwater on this, but otherwise I'll translate afterwards. So.
Good copy.
Okay. I'm going to start with Zulu 1. Zulu 1 is going to be the liftoff. Zulu 1 complete.
Okay, so basically I was just saying Zulu 1 is going to be the liftoff. And then I took the sample in this Falcon tube and I said Zulu 1 complete. And so the folks that are on the surface, they're able to communicate back down to me and say, okay, good copy, we got that. And they're writing that in their notebook so that when I bring the samples back up to the surface, then they know which sample correlates to which kind of mat and what area we are sampling in. So that's what it looks like for the Falcon tube sampling. That was specifically for Marisol, and she'll talk about that in a little bit. Here's another type of sample that, I think really shows how beautiful this mat is. So this is a cut sample from the mat that's laid on its side, and it kind of looks like a piece of lasagna. You can see all of the layers in this mat, and those are the different growth layers. And so at the beginning of this talk, I was telling you we went to Tahoe and we were taking samples with Jello because it mimicked our study species. And so maybe now you can tell why that Jello was a relevant option for practicing our samples, because they are quite gelatinous and fragile, these mats. And so when I take the samples up to the scientists on the surface, they can then look at them in the lab. So here's Marisol looking at the samples under a microscope and making all of her wonderful observations that she does. So now we're going to hear from Marisol about her work here and the questions she's asking and awesome results she's finding. So here we go.
Thank you, Abby, and thank you all for joining us. So my name is Marisol Juarez Rivera. I'm a postdoc at the University of New Mexico, and I consider myself a geologist or a geobiologist. I am a UC Davis alumni. I did my undergrad and master's in the Earth and Planetary Sciences department with Dawn, who you'll hear from in a little bit. So I'm a scientist here and a dive tender. I help with the air distribution to the divers below. And I became interested in Antarctic research because I wanted to know what the role of oxygen in shaping these microbial mats or biosedimentary structures was. But over the past couple of years, that's shifted a little bit. And I'm interested in studying the disruption due to liftoff mat and how the microbial mats recover from that. So one other interesting thing about Lake Frixell is that it's a lake that is super saturated with gases. So on the left-hand side, we have a transect of dissolved oxygen. The black line is what you would see if the lake was in equilibrium with the atmosphere. The blue line is what oxygen is with depth. So there's so much oxygen in this ecosystem. Microbial mats are producing some of that oxygen. So on the right-hand side, we're actually seeing a microbial mat. And if you can spend a couple of minutes looking at it, you're actually seeing that there's a million bubbles within this mat. So the mats are producing oxygen and they are trapping gas bubbles that include some of the oxygen. And as Abby already sort of alluded to this before, the bubbles raise the mats and it deforms them and lifts them off of the lake floor, which is what we call lift-off mat. So this is a video of another video of the benthic floor. So the distance between the scale bars is 10 centimeters. So this is mat that would have otherwise been relatively flat on the lake floor, but because there's abundant bubbles, it's being lifted off of the lake floor into these really complex morphologies. In a little bit, we're gonna pan up and look at the underside of the ice. And what you're seeing is a lot of the mats are actually floating into the water column and getting trapped at that ice-water interface. Sometimes some of those mats will freeze onto the ice cover and they will be exported from the lake. So we can subdivide these mats into different types. So we have non-lift-off mat, the lift-off mat, the mat that goes up right under the ice cover, and mat that becomes exported. So it's a really dynamic system. And some questions that I'm interested in is how much of this carbon is getting removed from the lake, and how does this ecosystem recover? So what I'm showing you here is the amount of organic matter for different mats with depth. And we know that it changes. So in order to try to get a better estimate of how much biomass is being moved within the lake and out of the lake, we need to study mats at different depths and understand their composition. And when we sort of take a step back and think about the history of Lake Frixell and how this process has happening through time, we have some information that shows us that it has changed through time. So this is a graph of elevation on the y-axis. And then in gray, we have the thickness of the ice cover. And in green, we have the liftoff zone where this process is happening. And so from the 1980s up until a couple of years ago, we saw a change in the ice cover, but we saw a really big expansion of the liftoff zone. And if we turn those depths into a surface area, we had a doubling in the surface area where this process is happening. So there's so much changing. And again, it's a very dynamic system, and we're trying to understand what drives these changes and what the effect is on the ecosystem within the lake and out of the lake, because that's a lot of yummy organic matter that's getting exported. So another question is, we have these liftoff mats, they get removed from the lake floor, and then that ecosystem gets recolonized. So how are the steps that bring it from liftoff into a recovered system? So the image on the left, you might be able to see a little bit of difference in the color in the bottom. So we have more yellowy areas, and right underneath that liftoff mat is more tan. And so the idea is that the yellow areas, there was mat that left a previous season or a couple of seasons, and now it's being recolonized. And maybe in the future, it'll look again like the mat on the right side. So to try to test or see how the microbial community is changing, we collected lots of samples, and you see there Abby collecting some of these samples, and we'll characterize the microbial community. So lots of exciting research happening, and how this gets preserved in the rock record. Tyler will be able to tell you about this just coming up.
Great. Thank you, Marisol. I'm Tyler Mackey. I'm an assistant professor at the University of New Mexico, and I am a UC Davis scientific diver and a research associate there. Our project, as Abby mentioned, is B047, and I'm a co-PI on that with Dawn. And a lot of my work here is motivated by trying to understand how these microbial ecosystems are preserved in sediments, how they, how they can be used to understand changes in the system through time. And a lot of my objectives for this season were to sample for those records of lake history, but then also to put in a long-term experiment to think about and interrogate how the mats themselves respond to decreased light, what their sensitivity is to light versus water chemistry in this lake system. So giving a little bit of another tour here of the lake bottom. As Abby showed, there are dramatic changes with depth. As you are below the liftoff zone, you have these nice intact microbial mats that are growing on the lake bed. You come shallower, there's more light, more photosynthesis, more dissolved gas. Things are tearing up off of the bed. But if you go even shallower than that to the moat, this liquid water that forms seasonally around the edge of the lake, you have microbial mats that are growing there too in a very bright environment. And the microbial communities that are there vary based on these microenvironments, but the composition of the organic matter itself also changes. And together with Marisol, I have some students who are looking into these mats and seeing how the composition changes, how we can use that as a metric for rates of photosynthesis and changing environments through time. So in this plot, I'm showing carbon and nitrogen isotopes. In the lighter pinks, we have moat mats that are in those liquid water highlight environments. And then in the darker colors, we're going into deeper water settings, showing these trends in the composition of the mats that, if they're preserved down core, might give us some indication of how the system is operating and changing through time. So lots to dig into there. But one of the things that we know about this lake is that its level has changed through time as well. And just in the last 30 years, we've seen slight declines in water depth, followed by increases in water depth. And so what we're seeing right now in the lake is just a snapshot of time. And by looking down core, we can try to think about what is representative. How are the patterns that we see in relationship to how the lake was decades ago, centuries ago, millennia ago. So I've been taking a lot of cores through these sediments, freezing them, bringing them back to campus to dissect and investigate on a finer scale. But shallower than like 9.1 meters in our 9.5 and 10.2 meter cores on the left, you see those beautiful laminations representing multiple years of accumulation of these microbial mats. On the right side, you see just a few lamina growing above sand, and that we're interpreting as one of these regrown surfaces that may have delaminated and torn off a couple of years ago, and now is regrowing into the mat morphology. So one of the questions that we're really trying to ask here is, we have this beautiful signal of shallow versus deep photosynthesis, but how much of that actually ends up in the rock, in the sedimentary record here? Can we recognize shallow water communities, or do we have a bias in the way that we interpret environments from these cores? And much like Marisol is asking how much is exported as far as nutrients go, I'm interested in how many of those nutrients actually stay in the lake. So are we losing nutrients that may have been fixed into the mats in the shallow water? So some of the longer term experiments that I've been doing in the lake, in part also through the long-term ecological research network, are putting in shade structures that will stay for hopefully 6 years at the bottom of the lake. Here are the frames in the field of view that will be covered by mats to block out, not mats, shades to block out half of the light on the mat that it receives from the ambient environment, to really test whether the features that we're seeing changing with depth are due to the ambient water changes or changes in the light that the mats are receiving, and also looking at how sensitive these ecosystems are to disruption due to changes in lake level. So it'll keep us busy for a long time moving forward, and looking forward to seeing how these experiments show us changes in the mat system through time. So with that, I'm going to pass it off to Dawn here.
Hi, I'm Dawn Sumner, and I'm a professor at UC Davis in Earth and Planetary Sciences, and I consider myself a geobiologist. I'm the team leader for event B047 and responsible for bringing the group together and for getting the funding to be here and dealing with a lot of the logistics. I'm really interested in Antarctic lake ecosystems because they're mostly microscopic. So it's a little bit like going back into Precambrian time, when you didn't have any of the predators or the food webs were really just microbial. And they're hundreds of millions to billions of years of evolution, so the organisms aren't the same. So it's only a little bit like going back to Precambrian time. For this project, my objective is to install an experimental system to explore overwinter changes in oxygen and the redox state of the mats and how that relates to microbial metabolisms. And that's to try to understand how these ecosystems go from being anoxic to oxic with the onset of photosynthesis when we first get light. And for me, that's related to trying to understand how we accumulated oxygen on Earth about 2.5 billion years ago. So I've worked a lot with microbialites in the rock record, which are these microbial ecosystems that are lithified and preserved. And so we have this honeycomb mat here with these ridges and pits that Abby described and Sol has also talked a little bit about. When we sample those and cut them, we can see the annual lamina. And so here, this dark outlined area outlined with minerals represents 1 year, and then the streams that flow into the summer bring mud in and they create these lamina. So this is another year and a third year here. One of the interesting things we've seen associated with the liftoff bubbles is that the lamina are a whole lot thicker. And so there's several lamina in this really thin area here. But 2 years ago, there was much more accumulation of organic matter. And then again in this year. And the way photosynthesis works is you have more light, you can do more photosynthesis, you produce more biomass, and you produce more oxygen. So we have loggers that record the amount of oxygen, and the colorful lines at the top here are representing the amount of oxygen just above these microbial communities at different depths in different places. And then the zigzaggy line at the bottom represents the amount of light that's coming through the ice. And so this is from 2003, and you can see the daily cycle coming through here, and you can see the daily cycle also in the amount of oxygen. That's that coupling between the light and the oxygen. And then this is going into February, which is the fall in Antarctica, and you can see the amount of light really declining and going down. And in some places, the amount of oxygen above the mat is also going down. As Sol pointed out, there's a lot of oxygen in the water, and that's keeping the oxygen high here. One of the interesting things is we have this really high peak where there's a lot of transparency in the ice. And so even though the sun was getting lower in the sky, more light was getting into the water. And we think that this is one of the times when there was so much photosynthesis that you're getting a lot of bubbles being produced and inducing a lot of the liftoff. So we have this correlation between photosynthesis and oxygen. This graph shows a similar thing with the daily changes in light under the ice here, but this time we're going from winter into the spring. And some of the mats are showing they still have some oxygen, and they're showing a lot of variation. There are a lot of physical processes that are mixing it as well. But down at about 10 meters and 9.5 meters, this orange line down here is showing there's no oxygen at all. We know there are photosynthetic organisms there, but they're not producing oxygen yet at this time. And so there are these things that happen. There's some hydrogen sulfide in those layers, and the hydrogen sulfide can inhibit the photosynthesis by the cyanobacteria, which are one of the main organisms in this lake. However, there are organisms that evolved more recently, some of the algae and diatoms in particular, that are capable of doing all sorts of different things. And it turns out that at these depths, the diatoms are super important. And as team leader, I get to invite people to join the team. And Elisa is an expert in diatoms that live in these strange conditions. So she's going to talk a little bit about them next.
Hello, my name is Elisa Merz. I am a postdoc at the University of Constance in Germany. I am a biogeochemist, and here on this team, I am also a scientific diver. So the cool thing is I get to use both the scientific and the diving skill, combine them, and then be actually able to see underwater what I'm working with, and also select what type of samples I want. My objective during this expedition was to describe and understand how the benthic diatoms thrive in this dark sulfidic environment. And here we see the microbial mats down at around 10 meters depth, which Abby had shown before. Diatoms are unicellular eukaryotic algae. They're really important as primary producers, as they produce 20% of the global oxygen. So they're really important in most aquatic habitats. However, it's really interesting that exactly at this depth, where there is very low light and a lot of hydrogen sulfide, these diatoms seem to be the dominating organism. And that is what a diatom basically looks like. So here on the right-hand side, you see an SEM picture. You see that they're a little bit boat-shaped. And what I get to do is, during diving, collect a sample, which is called a sediment core. So that's a plexiglass tube I can push into the sediment. I put a stopper below and a stopper on top, and I get this punch-out core to take back into the labs at Frixel, so I can study the chemistry there a little bit better and don't have to stay underwater for a very long time. And here under the microscope, you can see that it's very brown, and that's the color the diatoms have because that's what their pigmentation is. And we also call those areas the golden meadows because of those diatoms. And very interesting is also that there's one morphotype of diatoms that we see in that community. So it's very dominant, but dominated by one species. And here is a picture of what my lab setups look like. So on the left-hand side, you see that sediment core with the beautiful brown golden layer on top. And I use tools which are called microsensors. So in that bucket, there is the sediment core. And here you can see that there is a few sensors simultaneously measuring. And the cool thing about microsensors is that they're non-invasive, they're fast, and you can measure online. So their tip is thinner than a human hair, which is really cool because you can measure at the same point multiple times over time, and that gives you a time series. And I've been mainly looking into the photosynthesis at the Golden Meadows. We get 4 micromoles of photons per square meter per second down there. So it is pretty dark. It is very sulfidic. And when you look on the left-hand side, right-hand side, sorry, this is an oxygen sensor, and the black one is a hydrogen sulfide sensor. And in the profile, the yellow lines are oxygen. So we have a little bit of oxygen in the water column, and then we have these really small bumps. And these really small bumps are actually oxygenic photosynthesis, which is oxygen produced by these diatoms. And once I turn the light off, this bump slowly decreases, which is due to respiration. So it is a true photosynthetic oxygen peak. However, now that I came down here, I am absolutely fascinated by this incredible photosynthetic diatom-dominated ecosystem. And the next step is to look what they're doing when there is no light, which is also the other half of the year, basically.
Awesome, thanks. I'm just gonna double check that we're still live and recording because
You are
I
still
just want
live.
to make sure.
You're still recording, I promise.
We still
Everything
there?
is going brilliantly. Yes. Yes.
Looks like we're in it.
You are. You're
Hi,
perfect.
Jesse. Are we still good?
Yes. I think you can hear me, hopefully.
Okay, great.
We
Okay,
can
I'm
hear
gonna keep
you.
going then.
You're golden.
Cool.
Perfect.
Just wanted to check. Okay, cool. Anyways, great. So yeah, thank you, team, for sharing all of your amazing projects. And I just want to highlight that science really takes teamwork. And like the species that we're studying, when you're in an extreme environment, things become a little bit more obvious adaptations and things. And so one adaptation we must have when working in a really extreme environment is extremely good communication and teamwork. So I'm just really proud to be working with this team with all the unique skill sets and personalities that make our team stronger. So this was a photo from a day where we were lowering very expensive equipment down the hole and wanted to make sure we didn't drop it. So it took a lot of teamwork and communication. But you just heard from everyone about all the different projects that they're working on. So I want to show you a photo of what our schedule looked like. This was our whiteboard on the left of the last week that we were at field camp, just trying to plan everything out, trying to figure out how all of our projects fit together, how we could make the workload efficient and manageable and safe, and really get all of the data that we needed. And so you don't need to read all of the things that are on the whiteboard, but I think just the sheer amount of writing in different colors really explains kind of the brain power that went into planning this. And then on the left is our team sitting together in that Lake Frixel hut where we have our daily chats and we do our risk assessments. And so every day before we went diving, we did GAR, which is green, amber, red risk assessment. It really just facilitates communication. So if you've never used this before, it's something that we teach in our classes as part of the boating and diving safety department. And it's just a great way to really facilitate making good plans, having good communication, and knowing what everyone's up to. So we're going to zoom in on that real quick. We did adjust it so that it better fit the needs of our Antarctic environment and the work we were doing. So you can see we have on there, we talk about plan, we talk about resources, we talk about our individual fitness, we talk about the environment, what's going on outside, the complexity of our plan. And this was January 18th, which was our last day in the field in Lake Frixel. And you can see all of those numbers that we came up with as a team added up to a total score of 30. And then down at the bottom, there is a kind of a gradient of green to amber to red, which has different number ranges based on what your risk score is associated with the color. So a 30 ended us in the red zone. But this was honestly a really, really proud moment for myself and I think the rest of our team, because it just demonstrates how honest our discussions were and how thoughtful they were in planning and how everyone was really able to contribute, what they felt an accurate score was for the risk that was associated with the work. So nothing really is too low of risk in Antarctica because there's so many different factors that affect your work. But we were basically able to take this score that was in the red, which would technically be a no-go, and we were able to talk about it and bring it back down to a score that we were all comfortable with by making adjustments in what our plan was and what our different roles were. I think we had some folks come out later in the day instead of being out there the whole day and things like that, just to make sure that everyone was happy and we still got our work done. Because at the end of the day, that's the most important thing. So when we were done with everything, it was time to get off the ice. So here's Tyler on his final dive in our ice hole, basking in what has now really become like a pool. The ice hole really melted because we're in like peak of heat and sunlight. And so it just kept on expanding. So the ice hole started off not even being able to really fit a person through it, right? So it started off as like a really small hole, and then it grew as they were melting it. And we tried to dive in the beginning, and it wasn't big enough. And then we had to keep making it bigger. And by the end of the season, it just naturally was melting out and got this big. So the ice on the lake was telling us it's time to go. Conditions were getting a little bit harder to walk out there. And so now we're back in McMurdo after a really great, successful field season. So with that, we have lots of people that we want to thank collectively for supporting this work here, including the folks on continent that have been crucial to our team success, Ian Hawes, Taylor Peters, and then the diving staff that are here and the general support group. And then we have all of our friends and family back home and those who are watching, our pets, very important. So hi to everyone that's watching. And then especially Jesse, thank you so much for facilitating this talk. Jesse runs an amazing program called Exploring by the Seat of Your Pants, and I've had the fortune to do lots of talks with him to classrooms across many different states and even countries. And so he has generously offered to help facilitate this talk so we could give it to all of you at UC Davis and beyond. And then, of course, we have our UC Davis teams back home, including, but not limited to, BML, CMSI, Earth and Planetary Sciences, and Boating and Diving Safety Office, who helped get me here. And then, of course, the other institutions that folks are part of, University of New Mexico, University of Constance in Germany. So lots of different people. Thank you so much for watching. Thank you so much for logging on and spending your time with us. And I think if people are willing to stick around or have time, we might be able to take a few questions as a team now. We can read them in the chat.
Amazing. Well, thank you, Abby, and thank you, incredible team. There are so many of you, and so many folks are tuning in, both live on StreamYard with us, 73 people in groups on YouTube. So a whole horde of folks tuning in, and already a bevy of great questions. So I will note, we've got 25 minutes together. If you guys want to share more questions, please do keep them coming in the chat. I'm going to try and bring them on camera as much as I can. So some really simple ones to get us started. How long are you underwater typically?
Sorry, Jesse, you said how long underwater are we typically? I think.
Yes.
Oh,
Yep.
There it is. There's a question. Oh, yeah. Great. So we try and keep our dive times to about 30 minutes, but it really changes on the task at hand. And the reason we try and kind of keep our dive times short and manageable is because it takes a lot of effort of the folks that are on the surface tending our dives. And so got two of our wonderful favorite dive tenders here, Dawn and Sol over here. They're all going to get in. Come on, Tyler.
Everybody in.
Yeah. And so it can, depending on the weather, it can be harsher on the surface than it is underwater. So we really want to manage everyone's well-being on the surface. And so that's why, 30 minutes usually.
Lovely. Thanks, Abby. We've got one similar and related. What's the temperature range at the lake? Like how cold, how warm are you getting when you're underwater?
The water temperature ranges from 5 Celsius to 5 degrees Celsius based on the loggers that we've had. It's usually about 2 degrees Celsius, which is really quite cold for the divers, especially if they're not moving around. Above the air, we go from about minus 15 Celsius with wind chill to, I think the high was close to 8 Celsius this year. So sometimes 8 feels like really balmy and warm, especially with the sun shining.
Listen, minus 15 sounds warm compared to Newfoundland. It's like minus 25 with the wind chill here, and we had like a 3-foot snow blizzard. So there you are. We got some more technical questions, and I'm really glad we're getting some technical ones. So Estella wanted to know what kind of microbial communities are present in the mats, and perhaps related, is there a midwater microbial planktonic community? So can you tell us a little bit about what lives there in the water?
Yeah, so the main biomass in the lake is created by a combination of cyanobacteria, which are photosynthetic bacteria, and diatoms. And depending on where you are with depth, the specific species and the proportion of diatom versus cyanobacterial biomass varies. And and then those support a whole host of other organisms, from some of the nematodes and water bears to a fairly diverse suite of bacteria. So sometimes in extreme environments, the diversity is quite low. But in Lake Frixel, our prior results suggest that we're at a moderate diversity. So the photosynthesis creates enough biomass to support quite diverse communities. The density of organisms is highest on the lake floor, and we think that's because they can cycle nutrients to keep living. But there is also a planktonic community that has been studied quite a bit. It's in some sense easier to sample because you can get water from a hole in the ice to study those, and there's a 30-year running long-term ecological research project that has been looking at how those communities vary seasonally and through time. And so our work is really focused on the lake floor to complement the other work.
What a fantastic and detailed answer. Thank you so much for that. Yvonne wants to know, is there an estimate or ratio of O2 versus CO2 produced in these ecosystems? No pressure.
There's net accumulation of organic matter, and to create organic matter, you're consuming carbon dioxide and producing oxygen. So we are in the midst of struggling with how to calculate how much oxygen comes in from stream water versus how much is produced by photosynthesis, and how much of it then goes away. And so no carbon dioxide. Well, so the only carbon dioxide that's being produced in the lake is coming from the decay of organic matter that was also already created by photosynthesis. So there's more oxygen being produced in the lake than carbon dioxide because we're removing some of that carbon in the organic matter. But we don't know the details of the balance.
I guess that's a reason to go back and find that answer out next time. Or Yvonne, join them on the field and see what happens. We've got a few more great questions in the chat, but please do keep them coming, folks. We're whipping through these, so there's lots of time together. Yulia wants to know, how long was your field season and what did you do for fun besides diving?
Sure. Our field season this time was approximately 4 weeks at Lake Frixel. We got started earlier than that, but there was some airplane mechanical and weather delays. For fun, we took a day off every week. So we sometimes just hung out at the hut. We did our nails and did some face masks. Other people took a walk to the glacier, or we read or stretched.
Fantastic.
I will say that I play capoeira, which is an Afro-Brazilian martial art dance, and I made a berimbau, one of the instruments, from things I found, and that was my most fun enjoyment.
Of course you did. Of course you did dance fighting down in the bottom of the world. That's awesome. David wants to know, you guys are down there in summer, it's perpetual daylight. What's it like sleeping when it's day all the time, when the night ever comes?
think we have all different techniques for dealing with that. This season was the first time I tried a face mask, which was an incredible thing for me. I found it very helpful for getting a little bit of darkness and sleeping that way. But other people had other techniques.
I made a tent within my tent of my sleeping bag liner. So it just covered my face and now blocked out a little bit of the light. And then I used another cover for my eyes. It's a struggle sometimes, but you get used to it as well.
It's a lot of work to stay on a regular schedule. So we always had a defined start time so that the days don't just extend into like 26-hour days, because there's a lot to do and plenty of daylight.
Fantastic. Thanks, folks. We've never had a question quite like that before, so I really appreciate it. On that note, one of the ones we got from our kids in our broadcast a few weeks ago, what do you bring down there to sort of keep yourselves excited or give yourself a little thrill at the end of the day? We've had this question a lot, so do tell.
I had 4 bags of hot Cheetos.
What'd you bring?
Coffee. Good coffee and a little espresso machine.
I brought chocolate, and I also brought a game for everyone to play, which was these like sticky hands that you get in a party bag, and then some coins, and then you have to get the coins with the sticky hands. Some friends of mine back home taught me how to play that, so it's a great way to entertain scientists in a remote field station.
Absolutely.
Definitely brought tea. Yeah, that was the main thing, really.
For me, the most important thing is the team, and so I get to bring the people that I want to spend time with and work with. I don't get to bring everyone I want to be here. They don't necessarily always get approved to come.
That's a beautiful, beautiful answer, and puts hot Cheetos into stark relief as an alternate option. So thank you very much for that. We had a question about what your biggest challenge was. You're down in Antarctica, you're diving in a lake, you're in this amazing ecosystem. What is the biggest challenge? And you highlighted that great sort of risk assessment. Maybe it's diving itself, maybe it's keeping sane. Anything you want to share with us?
Probably a dive compressor loss. We had our dive compressor, which fills the tanks with air for us, quit partway through, and there was heroic, valiant effort on the part of Dawn and Abby to get that working, but had to have another one sent out.
That was our biggest cause of delay this year. The season went extremely well, and so not have air for a few days for diving and having to do other science to be the biggest challenge is pretty nice.
Yeah.
I think also we have a little small vessel that we have to pull ourselves across the moat, which is the part of the lake around the edges that melts out first and becomes liquid water. And so we have this little moat boat system that we needed to start using once that ice melted out. And it's a pretty straightforward system. Like you just pull yourself across, but so many troubles with the lines getting tangled, and in the wind, it blows like away and things like that.
And we had an iceberg that
was in the way. So that was kind of like, that was actually a big part of our GAR risk assessment was like, how's the moat boat doing? Are we going to be able to actually get on the ice? That was a pretty big challenge, I would say.
So getting too warm.
Yeah.
And melty.
Yeah, melting.
The unexpected problem in Antarctica. We're going to finish up with one more question just for our research audience, which is, what do you want to do next? Are there research questions that are unanswered that you're excited to go back for? Are there entirely novel places that you can dive that you're keen on? Anything you want to share about what the future holds for epic diving expeditions in Antarctica, maybe B048 or whatever it may be?
Yeah, so this is a field season that was designed for future work. And so Tyler mentioned when he was talking that he's putting in shades to block the light, and some of those are expected to be in for 6 years. And so he's also part of the long-term ecological research program here, and his work is sponsored through that. And so there's 6 years of continuity for that. This project was mostly sponsored by a grant that I got from the National Science Foundation, and it's for an overwinter experiment where we block the light. And so we put in shades that completely block the light to stop photosynthesis. And we'll go back in October, November to sample and compare the gene expression and the chemistry of the water and the mats that could do photosynthesis and the ones that could not. And we did that at a couple different depths. And so that's going to give us a whole lot of information about how these ecosystems work over winter for the modern, and also help answer my question about how oxygen actually works when it's a trace gas, when you're first starting to accumulate it in a microbial community. So we have those two direct continuous programs, but then both Marisol and Tyler are putting in pre-proposals in 5 days for the next projects that will be in different lakes, following up on some of these ideas.
Fantastic. Thank you so much, Dawn. And I guess we'll just wrap up with, if there's any final message you want to leave our audience tuning in from. We've got folks from a bevy of universities, a lot at UC Davis, of course, people that might end up on expeditions like this in the future themselves. Is there anything you want to leave them on before we say a big farewell to everybody?
My farewell to the students in the past was that there are many ways to get to Antarctica and many jobs that make this work possible. And so we have folks that work in the kitchen, we have folks that work in the airfield, we have folks that work wherever. So whatever skill set you have, there is space for it in Antarctica. So there's lots of different ways to come down. And so that's pretty cool if you ever want to go, many different ways. But otherwise, yeah, just really grateful for the communities we have supporting us back home. It takes a lot of sacrifice on your parts back home to make sure that we are able to get here and sustain our work here. So big thank you to all those teams that are supporting us back there. So thank you.
What a beautiful answer, and a huge thank you to all of you for joining us. I know it's a lot to take an hour out of your schedule, or an hour and a bit, so we really, really appreciate the chance to hear from every one of you. For all our university audience and anyone else tuning in at home, thank you guys for joining us, and hopefully you take the chance to learn a little bit more about Antarctica, the amazing work that these universities and their teams are doing down there, and I hope you have a wonderful rest of your day.